The cells of our body use many different types of signal transduction pathways, and some of these pathways utilize G proteins. For instance, we know that the epinephrine pathway and phosphoinositide cascades used by the cells of our body utilize G proteins, and because of that, we call these pathways G protein coupled signal transduction pathways. So, what I'd like to focus on in this lecture is discuss how a specific type of bacterial pathogenic agent that infects our cells can actually cause the malfunction of G protein coupled signal transduction pathways, and that can lead to many problems, as we'll see in just a moment. So what exactly is this bacterial pathogenic agent? Well, we call it Vibrio cholerae, which is basically that agent that causes cholera in humans. So Vibrio simply means it's a rod-shaped gram-negative bacterial cell. In fact, the rod shape is actually curved, and so it looks like a comma. Now, gram-negative basically means it contains a very thin layer of peptidoglycan in the cell envelope, and so if we stain it with the color purple, with the purple dye, and then wash that purple dye away, that purple dye will not be able to remain in that cell wall, and so it will be washed away, and the bacterial cell will in fact appear pink under the microscope as shown here. So Vibrio cholerae are curved, rod-shaped, gram-negative bacteria that can infect human cells. And in the presence of oxygen, they will in fact use oxygen to produce ATP molecules in a process we call aerobic cellular respiration. Now, in the absence of oxygen, they will switch to fermentation to produce those ATP molecules. Now, Vibrio cholerae can only survive and grow under basic conditions. If we place them into an acidic environment where the pH is low, they will not be able to grow, and so eventually they will essentially die. And what that means is these Vibrio cholerae are in fact acid labile. And finally, as we'll see in just a moment, these bacterial cells actually affect the activity of G protein coupled signal transduction pathways, as we'll see in this diagram in just a moment. So before we look at the details of this mechanism of infection, let's actually discuss how these bacterial cells can make their way into our body. So let's suppose we're in some third world country where they basically don't have some type of filtration system that basically filters the drinking water. And so you end up drinking contaminated water or eating contaminated food that contains this bacterial cell. So eventually it makes its way into our stomach. Now, in our stomach, we have a very low pH, a pH of about 2. And so what that means is these bacterial cells will not be able to grow in our stomach. In fact, the majority of these bacterial cells will in fact die off, but some may actually survive. And when they pass into the small intestine, we know that in the small intestine, the pH changes from acidic to basic. So in the stomach, it's around 2, but in the small intestine, it's around 8.5. And this is the perfect pH for these bacterial cells to actually grow, thrive, and ultimately be able to infect the cells of our body. So what types of cells do these actually infect? Well, it's the intestinal cells, the epithelial cells found in that small intestine. So let's take a look at the following diagram. So let's say we drink the contaminated water that contains Vibrio cholerae. It makes its way into our stomach where the majority of them actually die off because they are acid labile. They cannot survive under acidic conditions, but a few of them actually make their way into the small intestine, and that's where they begin to thrive, they begin to grow, and they begin to infect the intestinal epithelial cells of that small intestine. So let's take a small section of the small intestine. Let's zoom in on this section, and this is basically what we get. So we have the lumen of the small intestine, and this is our Vibrio cholerae. Now, this is the membrane of the intestinal epithelial cell. This is the cytoplasm side, and again, this is the outside. And these are all the different types of membranes that membrane proteins that we might find in our membrane. So we have the 7-TM receptor, which is also known as the G protein coupled receptor, and it basically contains G protein, but because it's activated, that G protein has moved on and bound to adenylyl cyclase. So if we think back to the epinephrine signaling pathway, this is exactly what it looks like. So we have our 7-transmembrane helix receptor that basically is bound to the G protein, but when that ligand, the primary messenger shown in orange, actually binds onto the 7-TM receptor, that G protein, the G alpha protein to be more specific, actually detaches and goes on and binds onto adenylyl cyclase. And then the cyclase basically transforms ATP into cyclic AMP that goes on and binds and activates protein kinase A. And protein kinase A goes on and affects different types of enzymes and protein effectors. So let's suppose we have this bacterial cell. And so basically in step 1, what the cell does is once it makes its way into the lumen, it begins producing and releasing the cholera toxin we call choleragen. And so this is basically what it looks like in step 1. Now, what exactly is the structure of this particular toxin? Well, basically it's a protein that contains 6 subunits. 5 of these subunits are identical and the other one is different. So 5 of these units are known as the B chains and the other unit is known as the A chain. The A chain is shown in green and this is the enzymatic, it's the catalytic A chain that will cause its effect once it gets into the cell, as we'll see in just a moment. And these 5 binding B chains basically create this cyclical structure that ultimately is used to actually bind onto a special region of the membrane of the intestinal epithelial cells. So once inside the lumen of the small intestine, the cholera secretes the cholera toxin we call choleragen. This is a protein that consists of 2 types of chains, the A chains and the B chains, and they combine to form a hexameric structure as shown in this particular case. And this is the region that basically binds onto a special region in the membrane we call the GM1 ganglioside. So the choleragen uses its B units to bind onto the membrane sphingolipid called the GM1 ganglioside. So a ganglioside is basically a sphingolipid that contains sugar molecules attached onto that polar section. And so this is what the G1 ganglioside actually looks like. We have this lipid component inside the membrane, and we also have these 5 sugars shown here, which are basically pointing towards the outside of that membrane. They're found in the lumen of that small intestine. And so these 5 binding B chains basically go on and bind onto this GM1 ganglioside. And once they bind, an endocytosis process takes place in which the cell membrane basically invaginates and it engulfs this structure. And what happens is once that engulfing process takes place, this catalytic A chain is released into the cytoplasm. So once bound, the catalytic A chain moves into the epithelial cell of the small intestine via the process of endocytosis. Now, what happens once it makes its way into our cell? So let's suppose that our G protein coupled signal transduction pathway is actually turned on. And so what that means is this G protein is in its GTP phase. And so it is bound onto the adenylyl cyclase and it continually stimulates the cyclase to basically produce the CAMP, the cyclic adenosine monophosphates, which stimulate the pKa. Now, in the absence of this catalytic A chain, this G-alpha protein shown in red in the GTP phase actually has a built-in timer. And so it has GTPase activity, and that means it can itself turn itself off by using a water molecule to hydrolyze the GTP back into GDP. And remember, when the G protein is in its GDP phase, it is in fact turned off. But what happens here is this catalytic A chain moves on and binds onto this G protein in the GTP phase, and it catalyzes a covalent modification process. It basically covalently attaches an ADP ribose component onto this G protein in the GTP phase. And this covalent modification stabilizes the structure of this G protein in the GTP phase, and that ultimately traps this G protein in its active phase. So once this binding takes place, this G protein continually stimulates the adenylyl cyclase to produce the cyclic adenosine monophosphates, which in turn produce more active pKa molecules, which in turn carry out their function, as we'll discuss in just a moment. So the catalytic A unit, once it makes its way into the cell, it binds onto the G-alpha protein in the GTP phase, where GTP stands for guanosine triphosphate. And this GTP-bound protein is attached onto the adenylyl cyclase. Now, the A chain catalyzes the addition of an ADP ribose moiety onto an arginine residue found on this G protein in the GTP phase. And so this covalent modification of that G protein in the GTP phase stabilizes the structure of the GTP phase, and so this structure will not be able to turn itself off, and that traps it in its active state. And so if we move on to this region here, we see that the G-alpha protein will be trapped in the active state, which implies that it will continually stimulate the cyclic AMP production, which in turn stimulates the activity of the protein kinase A. And protein kinase A does two important things. Number one is protein kinase A actually phosphorylates the sodium-hydrogen antiporters, so exchanges. And remember, antiporters are these pumps, membrane pumps, that essentially utilize the gradient of one molecule to actually move another molecule against its gradient. And so because the pKa phosphorylates and deactivates the sodium-hydrogen antiporter system, this basically inhibits the ability of these intestinal cells to reabsorb that sodium back into the cell. And so what that means is there's a net flow of sodium outside of the cell, and the cell loses sodium ions to the lumen of that small intestine. Now, number two, pKa also actually opens up chloride ion channels. And so chloride ions basically move down their electrochemical gradient from the inside to the outside the cell into the lumen of the small intestine. And so what we see happening is there's a net flow of these sodium chloride ions, so sodium chloride salt, and it moves in a general direction to the outside of the cell. And remember, as these salt ions basically move into this direction, what will follow? Well, water molecules will basically follow, and water molecules will diffuse across the membrane and follow the net movement of this salt. So we see that the net loss of sodium ions and chloride ions from the cells drives the movement of the water out of the cells and into the lumen of the small intestine. And this leads to the loss of large volumes of water and electrolytes, and that's exactly why patients who have cholera basically experience very watery diarrhea. And essentially all this water will be lost through the small intestine, and this will lead to the dehydration of that individual. And so if the individual isn't treated, they will eventually die as a result of dehydration, as a result of loss of water and these electrolytes, sodium ions, and chloride ions. So we see that it's very important that our cells actually be able to function these signal transduction pathways effectively and efficiently, because if they actually malfunction, they can lead to very serious, very dangerous medical conditions.